Hi friends, welcome to my YouTube channel BAMU'S Biogenius. Today we will discuss about innate immunity. This topic is very important for UG as well as PG students. So stay tuned. Now when we talk about the history of innate immune system, the first scientist that comes in our mind is Russian zoologist Ilya Mechnikov. He was the first scientist to observe specialized cells which are acting against the microbial infection or acting against the microbes. And he was the first to identify the phenomenon of phagocytosis by leukocytes in a human blood. So that's why he is considered as a pioneer of innate immune system. Now the first question that comes in our mind is that what is innate immunity? We will define term by term. What is innate? Innate means inbuilt kind of a mechanism. Immunity means a freedom from. So this particular type of system gives freedom from any kind of microbial infection. Now, if we see the definition, it is a kind of disease-resistant mechanism which is rapid as well as non-specific, and it is given by or shown by our body against any kind of microbial infection. So this is the innate immunity. Now to understand a little bit better, we will have to see its important features. See, these are the important features of innate immunity. First one is it is a kind of disease resistant mechanism and it is present by birth in every organism including plants also. The another peculiar feature of innate immunity is that it shows an immediate response, and this response is non-specific. It is against the class of microbes that may be a that may be a kind of a bacteria or viruses or protozoans. So this is considered as a first line of defense against any kind of a pathogen. Then the last one is whatever the cells that are taking part in this innate immunity, these cells are present already in our body. So all these are the important features of innate immunity. Now as we know that this innate immune system is a kind of a defense mechanism. It creates different kinds of barriers against these microbes, and these barriers are physical barriers, physiological barriers, cellular barrier, and the cytokine barrier. We will discuss first physical barrier. Now, these physical barriers are meant for keeping away physically all these microbes, and this physical separation of these microbes is done with the help of two things that are present in our body, that is the skin and mucus. So first of all, we will discuss about the skin. You can see here skin is made up of three different layers: epidermis, dermis, and hypodermis. Now, the surface of the epidermis that is very special because it is made up of epithelial cell layers, and these epithelial cell layers are joined together with each other by tight junctions, and these tight junctions do not allow any kind of microbes to enter inside. Then another peculiar feature of this skin is that the various glands that are present in the skin that are responsible for keeping the skin surface acidic. The pH of the skin surface is near about 4 to 5, and because of that no microbial growth or microbes that can thrive on the surface of the skin. It has been also found that the skin secretes various kinds of antimicrobial peptides. These antimicrobial peptides are 50 to 70 amino acid long and positively charged. This positive charge of these antimicrobial peptides are responsible for binding with the negatively charged surface of the microbes. Even these antimicrobial peptides have a hydrophobic domain which is inserted inside the membranes of the microbes so that these membranes are destabilized, thereby killing these microbes. Another peculiar feature of the antimicrobial peptide cathelicidine LL37 is that it binds with the nucleic acid of the microbes, thereby stabilizing the nucleic acid. And that nucleic acid is presented to the PRRs so that the innate immune response can be generated. So this is how the skin or the antimicrobial peptide secreted by the skin helps in stimulating the innate immune system. So this is how that the skin is a kind of formidable barrier against all these kinds of microbes. Now the next physical barrier is a mucus. Mucus is present in the oral cavity. It is also present or it also covers the respiratory tract. For example, in case of the trachea, the inner linings of the trachea and bronchi that are covered with the mucus. Then a digestive tract. For example, in the stomach, the inner linings of the stomach that are covered with the mucus. Then the reproductive tract. So everywhere this mucus is present, and this mucus entraps various kinds of pathogens that comes in. So physically it avoids the entry of these pathogens inside our body. Another peculiar feature of a mucus is that in oral cavity the mucus is present or mucus is there, but this mucus is having certain kinds of enzymes, for example lysozymes, even antimicrobial peptides that are also present in the mucus. So these enzymes that are present in the mucus, for example lysozyme, is responsible for killing the microbes or pathogens. So this mucus is responsible for physically keeping away these pathogens and microbes from the body. Here, the viruses and fungi that are trapped in the mucin fibers of the mucus. So this is how that these pathogens are kept away from the epithelial linings of our body. Now we will discuss the second barrier that is known as a physiological barrier. Now this barrier includes physiology or certain kinds of biochemical molecules that are responsible for creating this physiological barrier. The first one is a saliva. We know that the saliva is secreted by salivary glands in our mouth cavity. This saliva contains various types of chemicals. One of them is, as we have discussed in the last slide, that it contains lysozyme. Lysozyme is an enzyme which is responsible for hydrolyzing the peptidoglycan that is present on the bacterial membrane. So because of that, that bacteria get killed. So this is how that this saliva that is a kind of a barrier against these microbes. Then another kind of a physiological barrier is acid. The stomach linings, or we can say that the stomach lumen is filled with the acid with the help of the epithelial cells. These are the specialized epithelial cells. These are called as a parietal cells or zintic cells. These are responsible for a secretion of acid in the stomach cavity. This acid is also responsible for immediate killing of the bacteria because the pH of this acid is about 1.5 to 3. So in this acidic environment, these microbes will not survive. Then another physiological barrier, that is a tears. As already we know that tears are also having these lysozyme. So any bacterial infection or any bacteria, if it tries to enter in our body through the eyes, the tears or the lysozymes that are present in the tears that will take care of it and it will kill that bacteria. So this is how that these type of biochemicals, they will serve the purpose of physiological barrier. So this is the second barrier that is against these microbes. Now the next barrier that is created by the innate immunity is a cellular barrier, and different kinds of cells that are the cells of the blood mostly that are involved in protecting our body. These cells are the neutrophils, eosinophils, basophils, monocytes, macrophages, mast cells, NK cells, that means the natural killer cells, and dendritic cells. So we will discuss one by one. So the first one is a neutrophil. The neutrophils are the most abundant kind of white blood cells that are present in the blood. About 50% of the white blood cells are the neutrophils, and these neutrophils have a very special, peculiar characteristic features. First is that neutrophils are the first to arrive at the scene of damage, and these neutrophils have a capability to leave the blood vascular system, and through these endothelial cells, these neutrophils, they squeeze and reaches up to the site of the injury. So this phenomenon is called as a diapedesis. Then another peculiar feature, a very astonishing feature of neutrophils is that these neutrophils are capable of netosis. Netosis means neutrophil extracellular traps. These are the traps that are, we can say that the neutrophils have a capability of vomiting out the chromatin material around that pathogen, and these chromatin material forms a net-like structure which entraps these pathogens. So this is known as a netosis. So the neutrophils are a very important part of the cellular barrier. Even these neutrophils have a capability of phagocytosing the pathogen. So all these characteristic features that makes neutrophils very important part of the innate immune system. Now the next one is eosinophils. Now these eosinophils, they are present in the peripheral tissues and on the lungs. These are helpful in clearing the parasitic infection. So we know that whenever we got an infection, the number of eosinophils that increases. So this is because to tackle the whatever the pathogens that are coming inside our body, to tackle those pathogens, eosinophils that increases. Now the next cells after eosinophils is basophils. Basophils are the granulocytes, and these are also responsible for tackling the pathogens. Then the next one is monocytes. Monocytes are the kind of cells. These are called as the largest type of granulocytes, WBCs, and these are responsible for forming or differentiating into macrophages and dendritic cells. Now these monocytes, when they after 1 to 3 days, they migrate to the respective tissue. They differentiates into different kinds of cells or macrophages, and these macrophages have a special role or a function that is known as a phagocytosis. So if these macrophages are present in the lungs, they are called as alveolar macrophages. If they are present in the kidney, they are called as a mesangial cells. If they are present in the liver, they are called as a Kupffer cells. And if they are present in the brain, they are called as a microglial cells. Now, all these macrophages are strategically located so that even if a kind of a bacteria or a pathogen that comes in, that can be phagocytosed. So This is the function of macrophages. Then the next one is mast cells. We know that mast cells, they are very famous for allergic reactions. They resides in a tissue where that particular tissue is in continuation or in vicinity of the external environment, so that whatever the pathogens that comes in through these external environment, that can be tackled. So these mast cells are responsible for inflammation and the release of chemicals such as histamines and heparins. So these histamines have a function in allergic reactions. So this is how that these mast cells are responsible for tackling with the pathogens. Then the next one is NK cells. Now these are the natural killer cells. These natural killer cells, they are responsible for killing the host cells if these host cells are found to be infected with the viruses. So this is how that these natural killer cells, they are present in the blood and they can move to the site of infections by the viruses. And the next one is dendritic cells. Dendritic cells got their name due to the dendrites that are present on its surface. These are the long projections. The long projections are helpful, or dendrites are helpful in tackling or mopping up more number of pathogens, so that they can kill these pathogens in large amount. So these are the dendritic cells are the most common in tissues which are in contact with the external environment. So again, here also these dendritic cells are strategically located, and so that they can tackle the invading pathogen. The function of these dendritic cells is that they phagocytose the pathogen, and along with that they have a special function, that is they are also called as a professional antigen presenting cells. So whenever a particular kind of a microbe that is engulfed by these dendritic cells, the parts of that microbe, or we can say that the antigens that are displayed on the surface of these dendritic cells, so that they can stimulate, they can instigate the adaptive immune system. So this is how that these dendritic cells, they take part in connecting the innate immunity with the adaptive immunity. So this is all about the cellular barrier that is there in an innate immune system, which is responsible for tackling or a first line of defense against the pathogens. Now let us see how this innate immune system works. See, whenever a person gets a infection, that may be due to the parasitic protozoan or a fungal or a bacterial or a viral, or if that person gets injured, the first line of defense, that is innate immune system, that comes into play, and it shows a kind of a physiological response. Now, this physiological response is in the form of a inflammation or a fever. Now we know that what happens in inflammation. In inflammation, redness of tissue that takes place, increase in the size of that tissue takes place, then increased blood supply to that particular tissue that takes place. Then in some cases, that is in case of a protozoan infection or a viral infection, that person gets a fever. So such a type of a physiological response is there. Now, why such a type of physiological response is there? What is the trigger for such a type of a physiological response? And the answer is large scale secretion of cytokines. These cytokines may be interferons, interleukins, chemokines, lymphokines. So all these different types of cytokines that are secreted by the cells of the innate immune system, and that brings about that physiological response. Now, what is the connection in between this infection, that is infection due to the parasites, and the physiological response, or this particular increased secretion of cytokines? What is that interconnection, or how these two are interconnected with each other? These two are interconnected with each other with the help of PAMPs and DAMs. So What are those PAMs and DAMs? That we will see in a next slide. PAMs and DAMs, these are the molecular patterns. These are the types of molecular patterns. You can see here PAMPs means the long form of the PAMPs is the pathogen associated molecular patterns, and DAMs is damage associated molecular patterns. Now, what is the pathogen associated molecular pattern? That means it is a parts of a virus, bacteria, or any kind of a parasite. Now, these viruses or a bacteria, they have a particular structural signatures, structural features that are associated with them and that are unique to these particular parasites. And that particular parts are called as a, or molecules that are called as a pathogen associated molecular patterns. Similarly, in case of a host, when this host tissue gets damaged, the parts of the cell that also produces certain kinds of molecular patterns. Such a type of molecular patterns are generated by the damage to the host cells, and that's why they are called as a damage associated molecular patterns or DAMs. So here these two types of PAMs and DAMs that are identified by certain kinds of cells of the innate immune system, and they have certain kinds of receptors on them. These receptors that are responsible for identification of these PAMs and DAMs. So that is, you can see here, the example here is the macrophage. Now you can see here that particular structural patterns of these particular microbes or a parasite that are identified by such a type of sickle shaped uh, receptors that are present on the membranes of these macrophages. So these are called as a PR receptors or pattern recognition receptors. So these pattern recognition receptors that are present on the membrane as well as they are present in the endosomes, or they are present also in the cytoplasm also of the macrophages. So whenever a particular pathogen that enters in our body, these particular parts of the pathogens that are recognized by these particular pattern recognition receptors. Similarly, if a epithelial cell, for example, here we have taken example of epithelial cells, host epithelial cells. Now, due to the infection, due to the say viral infection, this particular cell if it gets damaged, the ATPs that are present in the cytoplasm of this particular cell that are released. Now, extracellular ATPs, this is an unusual kind of a thing that suggests that there may be a damage to the host tissue. So these extracellular ATPs, they are treated as a TAPs, and these extracellular ATPs that are then recognized by these pattern recognition receptors, and they create an immune, or they start to create an innate immune response. So this is how that PAMs and DAMs that are related, or they are used by the cells of the immune system to stimulate the innate immune system. Now let us talk about the pattern recognition receptors. As I told you that pattern recognition receptors are a transmembrane protein. That means the proteins that traverses through the plasma membrane, and it has a domains, or it has a ends that are, one end is at the outside of the cell and another end is at the inside, that is in the cytoplasm. So it is more or less like this. That is, this is the extracellular domain, and terminal domain is the extracellular domain. It is also called as a leucine rich repeat. and this is because it contains more number of or we can say that the repetitions of the leucine amino acid. Then on the cytoplasmic side, it has a cytoplasmic domain which is highly conserved, and this cytoplasmic domain is responsible for taking the signals from the outside to the inside. So the signals that are, or the PAMPs that are associated, or these PAMPs will associate with this leucine rich region of the extracellular domain, and because of that there will be a conformational change in the cytoplasmic domain, and this is how that cytoplasmic domain will take that signal downwards into the cytoplasm, and so that these macrophages can respond appropriately to the PAMPs and DAPs. So this is how that these pattern recognition receptors that are responsible for instigating a proper innate immune response against the PAMs and DAPs. Now, another important feature of this pattern recognition receptor is that these pattern recognition receptors, in some cases, they form dimers, and then only they can transmit the signals to the cytoplasm of the macrophages. Now, you can see here such a type of dimers are formed, that these dimers may be a homo or a heterodimers. And what is the significance of these formation of oligomers or dimers is that they can enhance the process of signaling. A very small amount of a PAM that can give rise to, or that can stimulate the response on the cytoplasmic side, and that particular response is transmitted inside the cell. So this is how the oligomers that helps in identifying that particular type of a PAM. So this is how that the pattern recognition receptors that are very important in stimulating the macrophages so that they can produce different types of cytokines, and these cytokines then are responsible for the physiological response, or the instigating or stimulating the first line of a defense. So this is all about the innate immune system. Thank you very much for your patient hearing. Please subscribe to my YouTube channel BAMU'S Biogenius for latest uploads, or like my videos and don't hesitate to comment. Thank you.